Have you ever found yourself pondering the mysteries of cell culture? What it is, how it works, or perhaps even the difference between primary and secondary cell cultures? Well, you're not alone. Today we're going to dive into these questions and more to help unravel the complex world of cell culture. Cell culture, at its most basic, involves the growth of cells under controlled conditions. Typically, this occurs outside their natural environment, in a lab. Now, there are two main types of cell culture, primary and secondary. Primary cell culture comes directly from the tissue of an organism. You could say it's the first generation. These cells are complex, diverse, and closely mimic the physiological state of the organism. However, they have a limited lifespan and can be tricky to maintain, as they require specific and often demanding conditions to thrive. On the other hand, secondary cell cultures are derived from the primary culture. They've been subcultured or passaged, meaning the cells have been removed from their original culture and placed into a fresh growth environment. The advantage: these cells have an extended lifespan and are easier to handle. The downside: they may lose some of their original physiological properties over time. Moving on, there are numerous types of cell culture media available, each designed to support different cell types and research applications. The right choice depends on factors like the specific cell type, experimental goals, and personal preference. One common type of medium is serum media, which contains growth factors and proteins that support cell growth and survival. However, it's not all rainbows and unicities. Serum media has its drawbacks, including variability between batches and the potential for contamination. So, how do you choose the right cell culture media? It's a matter of considering your cell type, the nature of your experiment, and any specific requirements your cells may have. And remember, what works best for one cell type might not be the best for another. As with any scientific endeavor, success in cell culture comes with experience and the knowledge of a few key tips and tricks. For instance, always handle cells gently to avoid damaging them, maintain a sterile environment to prevent contamination, and regularly monitor your cells to ensure they're healthy and behaving as expected. So, to recap, cell culture is a vital tool in biological research, allowing us to study cells in controlled conditions. Primary and secondary cell cultures differ in their origins, lifespan, and handling requirements, with each offering its own unique advantages and challenges. And the selection of the right cell culture media, whether it be serum or another type, is crucial for successful cell growth and experimentation. Remember, mastery of cell culture requires patience, practice, and a keen understanding of your cell's needs. So, keep asking questions, keep exploring, and most importantly, keep cultivating your curiosity. Because in the world of cell culture, every cell has a story to tell.